Hello, I'm Елена Swiderskaya and I'm going to introduce you our team, Dr. Ken Lane, Professor Yuri Korchev, and Sir Christopher Edwards, and our project, a new approach to understanding the role of tumor microenvironment in anti-cancer drug resistance. This is a joint project with Imperial College, and supervisor from Imperial is Professor Yuri Korchev. It's potential industrial case, and supervisor from the company is Sir Christopher Edwards. We have a long-standing collaboration with Imperial College. We published several papers and have a joint PhD student. I'm director of the Functional Genomic Cell Bank here at City St. George's. It's a unique resource with a big collection of mammalian cell lines, mostly melanocytes and melanoma lines. We have immortal human and mouse melanocytes. Majority of the cells have been established here at St. George's. We send the cells to researchers around the world, and some of these lines will be used in this project. Melanoma is a cancer with the most rapidly growing incidence in the UK. It is the most malignant form of skin cancer that is responsible for about 3/4 of deaths related to skin cancer. It occurs as early as age 15 and is highly resistant to cancer therapies. Melanoma is characterized by abnormal proliferation of melanocytes. And for melanocytes to become transformed, several clonal events should take place. Loci like BRAF, PTEN, CDKN2A have established importance in melanoma. There are several stages of melanoma progression: radial growth phase, vertical growth phase, and metastatic melanoma. And acquisition of vertical growth phase properties is important for cells to enter dermis rather than grow in epidermis where melanocytes are, and then proceed to spread. In melanoma, like in many other cancers, the tumor microenvironment plays a critical role in driving anti-cancer drug resistance. Cancer cells, through their altered metabolic activity, secrete various metabolites and reshape the tumor microenvironment, influencing not only tumor progression, but also its sensitivity to treatments. We propose to use novel biosensors based on scanning ion conductance microscopy to monitor and control cancer microenvironmental factors and to evaluate their involvement in anti-cancer drug resistance. We have already performed studies and published data on 3D topographical imaging, extracellular pH, as it has been shown that acidic pH is linked to cancer progression, measurements of reactive oxygen species ROS, and cell stiffness in living melanoma cells. So the objectives for this project are the following. First, to establish 3R melanoma cell line models where various tumor microenvironments will be controlled and monitoring the local extracellular concentrations of tumor metabolites like pH, ATP, ROS, oxygen, lactate, and continuously readjusting them to specific tumor microenvironments. Second, in melanoma cell line models with various defined tumor microenvironments to evaluate changes in gene expression profiles to anti-cancer drugs and monitor the cell responses to treatments. Third, to evaluate the effect of anti-cancer drugs in melanoma cell models representing different stages of progression and various tumor microenvironments, focusing on changes in melanoma cell phenotype, pericellular pH, stiffness, intracellular ROS, and drug resistance. So here at City St. George's, we will perform cellular and molecular techniques, and Ken, another supervisor from City St. George's, will introduce some of them. Yuri will tell us more about scanning ion conductance microscopy developed in his group at Imperial, and Sir Christopher will discuss industrial part of the project. I also would like to say that PhD student who worked on a similar project, also supported by MRC, has completed and passed her viva this year. Emily passed her viva in April with a straight pass. She has 4 published papers so far, and 2 papers are in preparation. Her project was industrial case in collaboration with Imperial and ICAPIC.
Hello, I'm Yuri Korchev. I'm a director of nanomedicine laboratory at Imperial College. As Elena mentioned, the tumor go under several changes during the growth. Cell change their gene expression signature, are characteristic of cell in extracellular environment that lead to drug resistance. We developing a different nano tool to understand the link between these changes, applying scanning eye conductance microscopy where very high sensitivity probe can be used to monitor on live cells these changes, and that would help us to understand drug resistance and cancer treatment. Nanosensor we're developing are based on the glass nanopipette, which is very easy to prepare. First of all, attaching to scanning probe microscopy, like ion conductance microscopy, we can grab a very high resolution 3D image of a live cell and see changes in time, like this topography of melanoma cells, or changing in mechanical characteristic, follow with topographical changes and effect of drugs, how they affect. As lots of anti-cancer drugs are used to target cytoskeletal structure to change their characteristics. The probe could be modified further to introduce the pH sensor. So the particular membrane inserted into the tip, and it's a very simple procedure, and we can detect the release of proton from the cell. And you can see from melanoma cell, they're very heterogenic, and some cells produce a very large secretion of proton with other not. So these changes could be monitored. And most important, we can then grab a particular cell or using a specially designed nanotweezers for uh, gene expression analysis, which could be taken to the PCR. We already found the single cell changes during the cell, and the modification and differences is dramatic, like protein involved in genes involved in the proton secretion are quite dramatically vary within the culture. What is important is probe also can be modified with carbon to detect the ROS or oxygen. So we can do it intracellular, or even we can measure ROS production in the whole animals and see the effect of drugs like doxorubicin within the cancer and penetrate deeply into that. So these tools provide quite a variety of that, and the aim of this project apply this tool and maybe develop further sensor required for that to understand the link between gene expression and functional characteristics of the cell.
Hello, my name is Dr. Ken Lane, and I'm a reader at St George's, University of London, and I'm a third member of the supervisory team. I work within a broad area of genomics and gene expression and have a technology-based focus within these topics. In particular, I work with omics techniques, elucidating genetic variants and gene expression using advanced omic techniques like microarrays and next-generation sequencing using both Illumina and Nanopore platforms, as well as the more traditional real-time based approaches. Concomitant with this focus is, of course, the bioinformatic and data analysis necessary to understand phenotype, genotype, and gene expression relationships underpinning such data. The PhD will inevitably encompass many aspects of these techniques and technologies. Within my supervisory contribution, I will, of course, play a role in guiding and supporting you in these aspects.
My name is Sir Christopher Edwards, and I'm the chairman of iCapic, a technology company. I'm delighted to give this short piece in support of this important MRC project grant application. It's vital that we take forward our world-class technology. In the past, we have not done it all that well. If we take, for example, 3 Nobel Prizes for CAT scanning, MRI scanning, and also for monoclonal antibodies. We had Nobel Prizes, but we didn't take them forward from a technological point of view. So I hope we can do better. And this project is an important way of linking the basic science of cancer to this modern technology. In particular, It means that you can use scanning ion conductance microscopes to then look at the single cell and to look at the phenotype of that cell and compare it with the genotype. And so in relationship to a specific project, we can look at the cell stiffness here. Cell stiffness of a cancer cell is key to whether or not it metastasizes. If you can relate that to the genetics, you can potentially make a major advance. So I hope very much that you will be supportive of this grant application. Thank you.